Everyone. Welcome to the second session of the first day of this international conference. In this conference, we have altogether one plenary lecture and two invited talks. The organizers have reshuffled it. The first talk will be by Professor Wim Dehem from Belgium. So, since we are already delayed, Professor Wim, can you hear me? Am I audible?
Professor yes, Wim? I can hear you. Can you hear me?
Yeah, sure. You are visible as well as audible well. Okay. So, for the audience, let me introduce yourself and then I will invite you for the lecture. For the audience, okay. So Professor Wim Dehem comes from Belgium, Department of Chemistry. He earned his PhD in the year 1988 from KU Leuven University with Professor Dr. G. Lebe. Then he moved for his PhD postdoctoral program from 88 to 90 with Professor Dr. A. Hassner. Then he moved for another postdoctoral fellowship from 91 to 96 in the group of Professor G. Lebe. He came back rather. From 1902 to 95, he did another postdoctoral work with National Fund for Scientific Research. Then in the 1994, he moved to UK for a 3 months postdoctoral fellowship with Professor Dr. J.F. Studart, Nobel Prize winner in the year 2016. From 96 to 98, he was director assistant, KU Leuven. 1998 to 2004, he was associate professor at KU Leuven. Then 2004, he became full professor, KU Leuven. Since 2009 to 2018, he was head of the division, Molecular Design and Synthesis, Department of Chemistry. He has many research interests, the most important one being novel synthetic methodologies in supramolecular chemistry, porphyrins and calixarenes. He has many honors and recognitions to his credit. I don't want to read out all those things. So with these words, I very humbly invite Professor Wim to deliver his talk. Professor Wim, please.
Thank you very much for this kind introduction. I believe you can hear me, and that at this moment the screen, I can see it. I'm actually on the screen. So I would like also to thank the organizers for giving me the occasion for actually presenting our recent work. It's from the last decade, more or less, on 1,2,3 triazoles. A lot of people have collaborated on this, and you see their names here on the screen. I may even have forgotten some of the more recent people. I also thank the organizers and the speakers for the flexibility in putting me a little bit earlier in the program because I have other meetings today. And I apologize to all of the people present for not being in person here. I would certainly have liked that, but my schedule was too busy. So these are the topics that I'm interested in. Some of them we will also touch today. My main business now is heterocyclic and medicinal chemistry, also involving natural products and fluorescing dyes. Supramolecular chemistry indeed was my interest at the beginning of my career, but due to funding issues, I have now rather moved to some of the other projects that you see over here. Okay, so what I want to present to you today are 3 different methods that we have been establishing in our laboratory. I will focus mostly on the third method, since we have only like 25, 30 minutes, so I have to go a bit quick, certainly when talking about the first two methods. And at the end of the lecture, I will give the results of the last years. So I don't think the click reaction needs to be introduced. It made triazoles very popular in heterocyclic and other chemistries. These click reactions normally are metal catalyzed. So copper or sometimes ruthenium. There might even be other transition metals involved. Some people like to work on metal-free reactions because that may avoid some of the side effects of metals, maybe toxicity or maybe other things. So organocatalytic triazole formation also has been a subject of interest. It's actually equally powerful and actually complementary to the click procedures. So, and of course, in introductions, it will always be stressed that this is more eco-friendly and so on. So we have published a number of reviews that are here mentioned at the bottom of the slides. So organocatalytic ways normally involve things like reactive enolates or enamines. And they're actually, you could say, a modification of some of the most early synthesis procedures of triazoles, namely the dimrods from, let's say, carbonyl compounds under stoichiometric amounts of inorganic base. So why would we be interested in triazoles? Well, if you see here, actually triazole, 1,2,3-triazole that is, could be seen as a bioregister of the transamide. And the dipole moments and the presence of hydrogen bond acceptors and donors are actually quite similar. And of course, amides have a totally different metabolic profile than the triazoles. And I don't have time to go into it, but in this review here, which was not ours, but of a couple of years ago, a case is made for substituting amides with triazoles, and this is often a winning strategy, keeping or even enhancing bioactivities. So this is our first methods that we published is almost 10 years ago in 2014. It was on the first page of Angewandte, so it got a lot of attention. It's a novel three-component reaction involving aldehydes, nitro compounds, and azides, bringing them together at relatively high temperature, almost reflux temperature in toluene. This led to tri-substituted triazoles. So I remind you that click reaction normally gives you di-substituted triazoles. And this was possible with a wide array of substituents R1, R2, R3. But this has all been published long ago, although it might be interesting to check out the recent review about the use of nitroalkenes that you see below the slides. This is a reaction that works very well. If you want to know if reaction is efficient, it often pays off to apply it to multifunctional substrates. Then you multiply your yields. So, for instance, if a reaction has 80% yields, having the reaction twice would give you 64, and having the reaction actually 4 times, as you can see here in the triazole, still leads to a 51% yield. So that means potentially at least per three-component reaction, it's around 90% yield in this case. Also an excellent way to functionalize porphyrins and potentially also making them water-soluble after saponifying the ester groups. This is intramolecular version. So we leave the concept here of three-component reaction, but we gain some, let's say, rate enhancements. So now we can use L-proline and 37 degrees, so body temperature, you could say, rather than room temperature. Although I don't know what is the temperature in your room at this moment. So that was published also some time ago. That's a clear entropy advantage. You get some interesting fused triazoles, also not readily available via the classical click reaction. So, and this is a more recent example here where we applied this to nitrochromines. So again, it's not exactly the three-component reaction, although we prepare also the nitrochromine in situ from nitroalkene and the salicylic aldehyde. And then after the formation of the intermediate, we add the azide to it and we do the usual way, getting a reasonable yields of the well, fused chromine, you could say, and with some fantasy, you could see some analogy to natural products, flavonoids like narangenin and camphor. So this is a second method in which very readily available compounds like acylated arenes, azides, could be alkyl or aryl azides, and one carbon fragment which is the DMF acetal. So they're brought together, and what we get is an acyl triazole. You see a couple of examples over here. This was published in 2016, at least our first publication, and it actually is part of a research line that we have on the reaction of enamines with azides. I can't go into the details. There's a review here in 2018 which describes a very rich chemistry that you can have. Triazoles are only one of the possibilities of the compounds that can be formed. So here we see how it actually proceeds. So again, it's a two-step, one-pot reaction. So in the first step, we condense the DMF acetal with the enolizable methyl ketone. This can, if you're in a hurry, it can be done by microwave in a couple of minutes, 25 minutes at 150 degrees, but it also goes nice and slowly overnight at 100 degrees. So if you don't have a microwave equipment, no problem. And then you can bring the temperature down again to 100 degrees, add the azides, and this cycloadds regioselectively and also eliminates dimethylamine. I don't want to go really into the details of the mechanism, but it's on this slide. So there's actually first a demethylation, a fragmentation of the DMF acetal. This functions actually as a base methoxide, and then you get the enolate. The enolate reacts with iminium salt and then eliminates a second molecule of methanol, and then the enamine is formed. And this could actually be isolated if needed, but of course we like to proceed immediately with the azides. And here you see the regioselective cycloaddition followed by the dimethylamine elimination, which goes fast here because the hydrogen is actually quite acidic due to the presence of the ketone. So we have here a number of examples proving that acyl triazoles are important. Earlier work was either involving Grignard reagents or other unwanted rather reactive species. So this is much more straightforward. So also here on the slide you see a number of examples. The yields are generally okay, so 70 to close to quantitative yields can be applied with metal organic complexes, also again with porphyrins, even sensitive things like nucleosides. AZT here was tried, so that all works. Again, as you can see, alkyl and aryl azides both work. Although aryl azides need sometimes a little bit more time to react, and that's due to the intrinsic reactivity towards enamines. So aryl azides are always more reactive due to the fact that the enamines are the nucleophiles and the azide then becomes the electrophilic components. So here we see an application to natural products. One thing to remark is that if you have acidic groups, like phenols or carboxylic acids, they get methylated in the process. And that's due to the methylating behavior of the DMS acetal. So that's one thing to keep in mind. Simple alcohols will not get alkylated, only the acidic OH groups of phenols and carboxylic acids. So this is a more recent example where we actually bought an enamine and applied it to nitrophenolazide, which is quite electrophilic, gives close to quantitative yield of a very interesting aldehyde, which you actually can see as a kind of stable and protected malon dialdehyde, or diazo, I should say, malon dialdehyde. Now, if you condense this with primary amine, and it works best for alkyl primary amines, what you get is a rearrangement. Actually, it condenses with the aldehyde and then rearranges, forms a new imine which hydrolyzes to nitrophenylamine, to nitroaniline, and a very interesting compound which is on this slide called 26. As you can see, this is actually working very well for alkyl amines. For the anilines, actually, the reaction is in equilibrium, and we cannot push it to quantitative yield, so it stays somehow around 50%. Now we're also applying those aldehydes to different kind of reactions. It was published 2 years ago. And this is, I think, our main course for today, that our third method, which is actually a very general one, and also using very simple starting materials. We already published quite a lot of papers on this, and we just published in 2021 an account on the reaction which we have called triazolization of ketones. So if you have a ketone, you can make, in general, triazole as long as there are two enolizable hydrogens. So this is how it works. Again, it's a three-component reaction. But, one component is always the same. That's the nitrophenolazide, and we use only the two nitrogens. You could say it's not atom economic, but actually you can reuse the aniline if you would want, because this aniline is very readily converted to the azide by diazotation and adding inorganic azide. So what do? We add an enolizable ketone, we add an alkyl amine. It works again best with alkyl amines, and in toluene and heat, sometimes a small amount, a catalytic amount of acetic acid helps to speed up the reaction. Again, an enamine is an intermediate. Now, this leads again to an aminotriazoline, and the aminotriazoline, we can't really go to the details of the mechanism, but it rearranges, and then it will expel nitroaniline at some point. And in the process, again, we get tri-substituted triazole, again, complementary to known click chemistry. So this works well for a range of compounds, including indeed 1,4,5 tri-substituted fused triazoles, which we can't make from the corresponding acetylines because they're not very stable. And then any amines containing different kind of functional groups, including secondary amines, sp2 and sp3 hybridized double bonds, even acetal. Are possible without cleaving the acetal. We can do the triazolization. And you can include stereocenters, like, for instance, amino esters. We didn't use amino acids because they're not soluble in toluene. And so this is why we used the esters. But as long as you control the temperature and the amount of acids, you won't have any racemization. This is a kind of busy slide, but it shows that it's possible to apply it to complex natural products that have either an amine function or a ketone function. And so it's possible to introduce the triazole into the skeleton, for instance, of triterpenes. And This has been applied in our earlier publication, but we have continued with that through the years. And for instance, we applied it to piperidons and got a compound which actually had an interesting anti-coronavirus activity. And it's a publication of 2018, so this is obviously not the COVID-2, the SARS-2, but another variety that causes the common cold. But we didn't think our paper would get that much attention later on, because now it has been, of course, highly cited. Okay, so this is an example here where we apply this to dendrimer chemistry. So again, multifunctional. You see the triazolization is really, really good. We push it, of course, with excess of ketone, but you can push it up to 73% for 8 different reactions occurring. So that's quite successful. It's also possible if you use, instead of an amine, ammonium acetate to get NH triazoles. Many of those have been present in pharmaceutically active products. And without going into the details again, you can see here that if you compare to literature procedures, this is very effective. So in literature procedure, we have to go through 3, 7, or even in some cases 13 steps. Of course, with not so good overall yield, whereas with more or less the same starting material, we can now prepare the compounds in 1 step and in a good yield. This is a recent relatively recent publication where we indeed went a little bit further with our natural products analogs. This is a betulinic acid analog. And again, it showed an interesting anti-coronavirus activity. We did modeling on it. Later on, it was tested also on the emerging, at that time, SARS-2 variant, but unfortunately, it didn't work for that. And the enzyme that we actually were targeting, it was seen that it was slightly different, and the pocket was a little bit more narrow, so the lead compound did no longer fit. And we are continuing to work on this. This is a publication where we describe the total synthesis of papaverine. This is a very short total synthesis and using triazoles. What we are doing is further triazolization on the acetophenone. Works very well, close to quantitative yields, including the acetal. And then we have a so-called Pomerance-Fritsch cyclization of the acetal into the electron-rich dimethyl aryl group. This also goes quantitatively in sulfuric acid. And then actually it's possible to, in the presence of veratrol, to open up this ring and to immediately have a Friedrich-Krass alkylation onto the veratrol. And this gives the papaverine in 3 overall steps. It's a very good yield overall, in fact. And the intermediate triazole or isoquinoline, in fact, can be ring opened and converted to a lot of different potentially pharmaceutically active compounds. So here we see another application of this chemistry where we build up CXCR4 antagonists using the substituted isoquinoline building blocks. And these are leading to very high activities in the nanomolar range. Here we see the so-called Combreta triazoles. Combretastatin is a compound that has been described as an anti-cancer agent due to inhibition of tubulin polymerization. So it's not a statin per se, so that's a totally different compound, but it's a different biomechanism. So it was known already before that the heterocyclic ring can take the place of this cis double bond. So, and these were compounds that were known already in the literature. So we looked at this, we made a whole library of compounds, and to make a long story short, our best compounds were the one that you see below on the right, 101 and 107, where we had activities of, well, a few more, 50 to 150 nanomolar in different cell lines. This work has not yet been published. So this is another recent paper that we published in Molecules. It's another three-component reaction, a new method, you could say it's method 4, in which we also start from acetophenones, but now the two other components are azides and sulfinic salts. It's copper catalyzed, and it's actually involving a Dimrod type of reaction. So, there's two catalytic cycles, and in fact, first the acetophenone is oxidized, and then you get an active methylene, which actually participates into the, and that's part D of the mechanism, it participates into the reaction. And you get, again, a tri-substituted triazole involving a sulfonyl function, which is kind of difficult to get in any other way. So this is some other recent publication where we involved triazolization onto a compound that already contained triazole. So this is a way to get bis triazole compounds. Worked quite well, in fact. So when you see here a couple of examples, it works actually both on cyclic and acyclic compounds. So, and that's a recent JOC publication. Here we see an application of the triazolization on steroids. Steroids very often have ketones, so we took advantage of that. And as you can see, the triazoles here are fused to the A or the C rings mostly. We built a library of molecules, and this was used for profiling of steroid receptors together with other collaborators from the KU Leuven. So here are, my other interest is fluorescing dyes. So this is a kind of a crossover into fluorescing dyes. One new analog of body pie that we are studying is BOPI. And here this was done by a triazolization of pyrrole of fused cyclohexanone. This only worked when the nitrogen was tosylated. We then remove the protecting group. We make an aldehyde, and this is now the starting material for the dye. And what happens next is that we condense this with a hydrazide. We chelate it with BF2, and this then gives a fluorescing dye, which can be made water soluble. That's another advantage of the triazole. You can methylate it and increase water solubility. So, and indeed, we get a water soluble fluorescing dye, which can, of course, target, for instance, in the cells, part of the cells that are negatively charged. This is another recent paper that we published, in fact, this year, and it has to do with the chemistry of azapurines. And here you see a couple of examples of azapurines that have appeared in the literature and actually are being used for different biological purposes. You could ask yourself how these compounds are prepared. There's actually a triazole first, or an imidazole, an pyrimidine first pathway, A and B, or you could, of course, elaborate an already made azapurine skeleton. So what we are doing is actually applying a kind of click reaction. It's a so-called arrested click reaction, in which we involve the copper intermediates into a reaction with sulfur and then methylate. So this gives the substituted compounds. It's sulfane, and then we oxidize it to a sulfone. This makes it a good leaving group, and then there's a condensation with amidine. So this is a rather short and general way of getting azopyrines. It also gives a substitution pattern, which you normally don't have with the other methods A to C. So it's again a little bit of a busy slide, but I think you can get from it that there can be variations at the alkyne parts. So this is introducing alkyl groups or aryl groups. That is normally not the case for these azopyrines. We can vary the azides with aryls, but also with alkyls. Sterile protecting groups could be present. Or we can vary the amidine. So also here we have different possibilities, mainly alkyl and aryl groups, but also, for instance, an amine is possible. So post-functionalizations are also possible. As you can see here, we have a dimethyl azapyrine. We again use our methods with DMF, that's method 2, via the enamine with DMS, and then we get another triazole at the 6th position. So this goes in good yield, and it's regioselective. And so also the aldol condensation, or you could call it also a Neuvenauer condensation, works. And we prove the regiochemistry with the X-ray structure that you see over here. And you could say that the compound 19 actually is a nucleoside analog or an acyclic nucleoside analog, so it was prepared via desylylation here. We're actually at the moment looking at the biological properties of this compound, but communication was already published in Organic Letters. Actually, you should have updated it. It's already published. Okay, so this is another paper that we had on azapyrines. It's actually method B, you could say, so pyrimidine first. So this is according to the classical methods. Here we focused mainly on CCR7 and CXCR2 activities. So we built a whole library, and we also published this year. So some of the compounds actually had quite interesting activities. It's also not very common to find something that is a CCR7 antagonist. So we actually made the first systematic STAR study for this class of compounds. So I think I used up enough of the time, and it's time to conclude. So we have seen several methods, and I think I spent the most time with the third method, which is the triazolization. I also presented some of the new work. So these are all, we are a synthetic lab, we work on new synthetic methods. So the more people use these methods, the better. So I urge you, if you are inspired, to apply these new methods. So, and I thank all the people that contributed to this. So these are the former members. There are also some current members still working on it. We collaborated with a lot of people that studied the biology of these compounds. Also, I should mention my good friend, Professor Bakulev from Yekaterinburg in Russia. It's a bit more difficult to collaborate with him now, but we still do it. And all the people that gave the funding. So this is a picture from the city center of Leuven. There's a statue here, and you can see this statue was also the statue that was on the first page of the Angewandte paper that led to our introduction into this field. It's a picture of a student and how the student can get the wisdom. So by reading the book, and I guess also by whatever is in that glass that he is holding. Okay, and with that, I like to finish. So I just put your attention also to a book that I wrote together with my good friend, Professor Bakulev, a couple of years ago on 1, 2, 3 triangles. Thank you very much. for your attention.
Thank you so much, Professor Wim Dehan, for your wonderful talk. Can we have a few questions?
Yes, please.
Yeah. So, the lecture by Professor Wim is open for discussion. If you have some questions, feel free and ask. Please come here and ask.
Hello, Professor Wim. Very nice talk, very interesting in it. I just want to know, do you have do you need dry solvent to do this kind of reaction? Because many, phenyl azides, they undergoes kind of reduction reaction, phenyl azide to just aniline type reaction. So do you maintain some dry condition for this kind of reaction?
It's not really needed to have an inert atmosphere. In some cases, like the triazolization, is actually, there's a molecule of water evolving. So 1 mole of water is actually generated. And this means that the water, of course, slightly inhibits because it pushes the reaction to the other side, to the starting materials. But we never take great care of, let's say, making everything super dry. So that is not, and I don't think really the water itself is going to reduce the azides, at least not under our circumstances. So we never saw these kind of products.
Okay.
So it's actually, normally our people actually working in closed vessels when they work at small scale, at the 100 milligram scale, and they work in open vessels, of course, with reflux, when we work on the gram scale, which works equally.
Okay, thank you so much. Thank you so much.
Mm-hmm.
Any other question, query, or clarification? If no more questions, let us thank Professor Wim for his very nice talk, full of exciting chemistry. Thank you, Professor Wim.
Okay, and thank you. I will stop sharing now.
Yeah, thank you so much.
Yeah.
Yeah.
Bye-bye.
Yeah, bye. Where that person has gone, who will connect to another speaker online?
Maybe they might have joined after that. Maybe you may announce either he may be there.
Oh, right.
I think they might have shared the link on the online.
So, Professor Konstantin Volcho,
Yes.
can you hear me? Yeah, am I audible or not?
I'm here.
Okay,
Do
fine.
you hear me?
Yeah, your voice is clear, but you are not visible. Okay? All right.
Okay.
No problem. So, let me introduce yourself first, and then I will very humbly invite you to deliver your talk. So, the second invited talk is by Professor Konstantin Volcho, who received his PhD in the year 1997 from Novosibirsk State University, Russia. Since then, he has been working at the Novosibirsk Institute of Organic Chemistry, Russia. Researcher Professor Volcho is focused on synthetic transformations of natural compounds, mainly monoterpenoids and their analogues, including catalytic reactions for the development of new biologically active compounds. The research interest of Professor Volcho includes the development of novel treatments against nervous system disorders, antivirals, and anti-cancer agents. Professor Konstantin has authored and co-authored more than 250 peer-reviewed papers. He is an inventor in 50 issued patents. He is a professor of the Russian Academy of Science. So with all my humility, I invite Professor Konstantin Volcho to deliver his talk on the topic development of new biologically active compounds based on monoterpenes. Professor Konstantin, please.
Thank you very much. Good afternoon, dear colleagues. At first, I would like to thank the organizing committee for the invitation to have a talk on such an interesting conference. I have never been in India, but I hope to do this in future. At first, let me briefly introduce ourselves. I work in Novosibirsk Institute of Organic Chemistry, which is a part of Novosibirsk Scientific Center, well known as Novosibirsk Akademgorodok. Akademgorodok is located 30 kilometers south of the center of Novosibirsk and is surrounded by pine forest. It includes Novosibirsk State University and more than 40 research institutes working in different fields, including physics, mathematics, chemistry, biology, medicine, and so on. And such a close location of research institutes is favorable for fruitful scientific collaboration. The main goal of our work is development of new drugs for several diseases, and we usually use various structural types of natural products as the raw material for the development of new biologically active compounds. By nature. Indeed, the number of commercially available synthetic compounds greatly exceeds the number of known natural products. At the same time, the amount of biologically relevant compounds among synthetic ones is not so large, while all natural compounds are certainly biologically relevant. Moreover, the importance of the search for the leader among the natural compounds and the derivatives is caused by significant chemical diversity of these molecules, including presence of chiral centers. So it is not surprising that approximately half of new small molecule drugs trace their origins to natural products. Among natural products, monoterpenes form one of the most widespread classes. These are natural compounds that can be formally considered as isoprene derivatives consisting of two isoprene units. Monoterpenes can be found in large amount in essential oils. Among most widespread monoterpenes are alpha and beta pinene, sricarine, and limonene can be mentioned. Very important that many monoterpenes can be found in nature in optically pure form, and sometimes both enantiomers are available from different sources. The oxygen-containing derivatives are called monoterpenoids. Several monoterpenoids demonstrate potent biological activity. For example, well-known monoterpenoid Mizoram is used in pharmaceutical preparations of the non-prescription group intended for the treatment of colds, rheumatism, relieving muscle pain, and so on. Another important monoterpenoid camphor tones the respiratory center and has a direct effect on the heart muscle. Nevertheless, the role of monoterpenoids in the development of new drugs is not agreed now. This fact can be explained at least partly by high chemical lability of many monoterpenoids and as a result often unpredictable reactivity. For example, acid-catalyzed transformations of relatively simple monoterpenoid, alpha-pinene epoxide, may lead to formation of up to 200 products. Some of them are shown here. Moreover, monoterpenoids often contain a number of functional groups with similar reactivity as well as asymmetric centers, which lead to additional difficulties for carrying out selective transformations. To solve the problems, we collaborate with several catalytic groups with the aim to find suitable catalysts for selective transformations of monoterpenoids to valuable products. And I will show a few examples. I would like to start with a Prince reaction of monoterpenoid alcohol minus isopolygon with aldehydes. This reaction leads to formation of corresponding type of bromines, and various catalysts, various acidic catalysts can be used. Dr. Gokul Bashir from Northeast Institute of Science and Technology, Assam, India, was first found that K10 monomelanite clay can be used as a catalyst for such transformation. We found that some products of this reaction demonstrated promising pharmacological properties. Thus, outstanding analgesic activity was shown by us for compound 1, which was more active and sufficiently less toxic than diclofenac. Important that the compound retains the effect for at least 24 hours after Perot's administration. Now we successfully finished preclinical studies for this agent. Note that the reaction is accompanied by formation of andesite and dehydration products. Moreover, only one stereoisomer possesses high biological activity. And this is why we try to improve the yield, and several different types of heterogeneous catalysts were tested in this reaction, including various types of clays by our colleagues from Institute of Chemistry of New Materials, Belarus, and our academy to Finland. The use of all these catalysts was quite effective, but the most promised catalyst for this reaction appeared to be halloysite nanotubes. The yield of target for stereoisomer was significantly higher with acid-rated halloysite in comparison with the results obtained with other aluminosilicate catalysts tested in this reaction. Also, halloysite is widely used as an efficient adsorbent. This material is very rare considered as a catalyst. So I will say a few words about this material. Halloysite occurs as small cylinders, nanotubes, that have a wall thickness of aluminosilicate approximately 10 sheets. The outer surface is mostly composed by silica, and the inner surface of aluminum. And acid activation of halloysite nanotubes leads to very interesting acidic catalyst, as we shown in our work. Substitution of halloysite nanotubes with boron trifluoride ecerate allows us to obtain fluorine-containing carbon with acceptable yields. Among these compounds, products with good antiviral activity against flu virus were identified. And based on biological data, hemagglutinin was proposed to be a target for these agents. Ketones can be used instead of aldehydes in such condensation too. The yields were lower than in case of aldehydes due to formation of dehydration products. Paracyclic compounds were obtained when cyclic ketones were involved in the reaction. These compounds demonstrated even better anti-flu activity with a very good selectivity index, which is a ratio of cytotoxicity dose to active dose. Selectivity index exceeds 1,500 in this case. It is a very good selectivity index for such type of antivirals. And again, hemagglutinin is considered as a plausible target. And now another our work is aimed at design of new antifungal compounds. Fungal diseases have a significant impact on public health, with over 1 million humans dying every year from them. World Health Organization published fungal priority precedents list on October 2022 to underline the urgent need for action. Azoles such as fluconazole and triconazole have long been first-line antifungals in fighting mycosis. At the same time, the emergence of azole-resistant strains is a serious problem. As it is known that monoterpenes and their derivatives often possess antifungal activity, we decided to combine azole and monoterpene fragments through piperazine link to overcome this resistance. Target compounds containing residues of recyclic, monocyclic and bicyclic monoterpenes were synthesized in acceptable yields. These hybrids demonstrated broad spectrum activity against all tested fundamental strains with excellent minimum inhibitory concentration values against both fluconazole acceptable and fluconazole resistant strains of Candida species. Compound with paramencil and pinenyl fragments demonstrated up to 100 times lower mix than fluconazole against clinical isolates. Very good, very interesting data and very fresh data. Another topic. Chemo and radiotherapy of cancer often focus on the induction of DNA damages. The ability of cancer cell to initiate DNA repair is a key mechanism for therapeutic resistance to chemotherapy. Therefore, targeting DNA repair enzymes can be used as a strategy to initiate the cytotoxicity of the currently available DNA damaging agents against cancer cells. Tyrosine DNA phosphodiesterase 1, TDP1, is an important enzyme involved in DNA reparation. TDP1 is believed to be responsible for drug resistance of some cancer. For example, TDP1 plays a key role in the removal of DNA damages resulting from TOPO1 inhibition by camptothecin, and its clinically important analogs such as topotecan and irinotecan, which are used in treatment of several cancers. Moreover, TDP1 is involved in the removal of DNA damages caused by other widely used anti-tumor drugs with different mechanism of action. It is important that the inhibition of TDP1 doesn't lead to significant toxic effects. Thus, inhibiting the activity of TDP1 can enhance the therapeutic effect of TOPO1 modulators, such as topotecan, for anti-cancer treatment without significant additional toxicity. We started this work in collaboration with Novosibirsk Institute of Chemical Biology and Fundamental Medicine and School of Pharmacy and Bioengineering at Kyiv University. Using a virtual screening of the bioscreen chemical library of natural products in the analysis, the soxabenzyl derivative of 7-hydroxycoumarin annihilated with cyclohexane ring was identified as a new TDP1 inhibitor with a C50 value of 5 micromoles. Preliminary molecular modeling against the TDP1 binding pocket indicated that the replacement of the aromatic substituent by bulky monoterpene substituents could improve the efficacy of the inhibitors. Then we synthesized a library of target coumarins and found that in all cases monoterpene substituted coumarins were more potent TDP1 inhibitors than the aromatic counterparts. Thus, methanol and geraniol containing coumarin conjugates with a C15 submicromolar range were the most active. Important that the conjugate at non-toxic concentration demonstrated clear synergetic effect, increasing cytotoxicity of camptothecin against M7 cell lines with a high level of TDP1 gene expression. At the same time, this compound didn't affect the activity of camptothecin against cell line with low level of TDP1 gene expression. This data can be considered as an indirect proof that the synergetic effect occurs due to the targeting of TDP1. More unambiguous proof that the synergy is derived from TDP1 inhibition was obtained using knockout of the TDP1 gene cell line, which was created using CRISPR-Cas system. It was shown that another TDP1 inhibitor, monoterpene derived, potentiated the cytotoxic effect of topotecan against the wild type cells, but not on an engineered cell line that doesn't express TDP1. Thus, the mechanism of synergetic action, at least for this inhibitor, is associated with the inhibition of TDP1. In vivo experiment revealed that coumarin-based inhibitor, being non-toxic, induced a significant increase in the anti-tumor effect of captopril on the Krebs to a side tumor model in dose-dependent manner. Thus, a high synergetic effect between TDP1 inhibitor and clinically used medicine in vivo experiment was clearly demonstrated. And the last topic of the talk concerns Parkinson's disease. This is one of the most common neurological diseases characterized mostly by motor disturbance. The percent of people affected by Parkinson's disease at the age of from 60 to 80 is around 1%. Among symptoms associated with Parkinson's disease are rigidity, bradykinesia, rest and tremor, and so on. And of course, cognitive and psychiatric complications. The main cause of the disease is an irreversible degeneration of dopamine neurons in substantia nigra of the brain, which leads to disturbed balance between cholinergic and dopaminergic neurotransmitter systems in the direction of cholinergic dominance. Among risk for Parkinsonism are genetic predisposition, approximately 10-15% of number of cases of disease. Drug Parkinsonism associated with the use of antipsychotics, contact with pesticides, some of which are neurotoxin, brain injury, and which is important for now, is viral Parkinsonism. Viral Parkinsonism is a consequence of encephalopathies caused by exposure to various viruses. One of the most striking outbreaks of viral Parkinsonism occurred after 1918 flu pandemic, and the COVID-19 pandemic is expected to lead to a sharp increase in cases of viral Parkinsonism too. There is no cure for the disease, and the goal for therapy is to provide relief from the symptoms by restoring the balance. The main medicine for Parkinson's disease treatment from the late 1960s and for the present day is levodopa. Unlike dopamine, levodopa is able to cross the blood-brain barrier and then is decarboxylated with dopamine, thus compensating dopamine deficit. Unfortunately, administration of levodopa causes several side effects such as nausea, appetite loss, involuntary movements, hallucinations, and so on. Moreover, prolonged use of levodopa give rise to on-off episodes, resulting in additional complications. And neurologists still fight to find the balance between clinical effectiveness and side effect of levodopa in each individual patient. Therefore, the research on new effective medication for the medical correction of Parkinson's disease remains very important. And I saw in the conference program that the first report, the first presentation today will be devoted to these important questions. During preliminary experiments, we found that several monoterpenoids synthesized by us simultaneously acted as agonist of the dopaminergic system and agonist of the cholinergic system, thus being promising for studying as anti-Parkinsonian agent, and we do such testing and found that among tested compounds, monoterpenoid diol was the most active. It is known that the neurotoxin MPTP induces Parkinsonian syndrome, reducing, for example, duration of locomotor activity in rodents. Diol-2 fully restored duration of locomotor activity and was as effective as levodopa. Compound 2 contains 3 assembled sensors and has 8 stereoisomers. We synthesized all isomers with high optical purity from corresponding anatomies of verbinons and carbones, and found that the absolute configuration of compound 2 greatly influences its anti-Dickinsian activity, from nearly full removal to sharp increase in the symptoms of the Dickinsonian syndrome. Thus, the most effective stereoisomer was identified. This isomer was called proteinin. It can be synthesized in 3 stages, starting from conventional Weberine. It demonstrated promising anti-Dickinsonian activity in all relevant rodent models and successfully passed first phase of clinical trials. This work is carried out with, in collaboration with LLC Innovative and Ecological Development, Tomsk, Russia. And during study of protein pharmacokinetic, its trans epoxide was identified as metabolite. This epoxide demonstrated high activity in TP mice model, has low acute toxicity, the 50 more than 1 gram per kilogram, and it is unusually stable in acidic conditions for epoxide. In addition to positive effect in MPTP, in acute MPTP Parkinson's disease model, we showed in cooperation with our colleagues from the Institute of Physiologically Active Compounds, Chernobylovka, Russia, that the epoxide demonstrated high neuroprotective activity against retinol-induced neurotoxicity by mitochondrial protection in vitro experiments. And I want to note that just retinol is responsible for one of the most important risks for Parkinson's disease, contact with agrochemicals. Okay. And the most important that in vivo experiments also confirmed that the epoxide protected mice from retinol toxicity, and we found that it restored distance and, by protecting dopamine neurons. And even more important, that as shall be shown in collaboration with the Institute of Biotechnology of University of Helsinki, Finland, that this epoxide demonstrated neurorestorative activity. In the MPTP mouse model, when the dopamine neurons were killed by it, the epoxide increased the density of dopamine neurons fibers in the striatum, and which highlights its potential to stimulate striatal reinnervation and thus halt disease progression. And this gives hope for development of really effective disease-modifying medicine. And I have shown some selected examples of our work aiming at the development of new biologically active compounds based on chemical modification of monotipenoids. Many other structural types of monotipene derivatives can be obtained by reaction of monotypics with carbonyl compounds, and more information can be found in our review and recent articles. And we are grateful to Russian Science Foundation for the financial support and all our colleagues for their valuable contributions. Thank you for attention.
Okay, thank you, Professor Konstantin. The talk is open for discussion. If you have some questions, you may ask. Any question from the audience? If no question, let us give a very big hand to Professor Konstantin for his exciting talk. Thank you, sir, for accepting the invitation to give your talk. Thank you so much on
Very
behalf
good.
of, yeah.
Thank you. Good luck with your conference.
Yeah, it's our pleasure to listen you online as well, no problem. Maybe next time you may come in person, okay? Thank you so much.
Okay.
Okay, thank you so much.
Thank you. Thank you.
Okay, thank you so much, sir. Bye. Okay, the last talk, which is actually a keynote address of this session, is by Professor P.M.S. Chauhan, who is from CDRI Lucknow. A lot of things have been said by different people about Professor Chauhan right from the morning, so he does not require much introduction. But for the audience, let me read out a few things which the organizers have provided me. Yeah, Professor Dr. P.M.S. Chauhan, he earned his PhD in the year 1984 from CDRI and joined as scientist in the year 1988. Professor Chauhan is included as 2% top world scientists according to Stanford University data. His H index is 48 and his citations are over 6150. So many things are provided to me. He has so many awards and recognitions to his credit, but to my knowledge, he is a person who has integrated chemists and biologists of the country. He is a man with a high academic acumen as well as high potential for organizing different events. So I have great admiration for Professor Chauhan, and I very humbly request him to deliver his talk on the topic Perspectives and Challenges in Drug Research, Design and Synthesis of Nitrogen Heterocycles as Novel Therapeutic Agents. Professor Chauhan, please.
Just tell me this. Sorry, this is next. This is next. What's up? What's up? Okay. Okay. This one. This one's next. This next. What's that? This next. And this one is. This one. Highlight. And this one's next. Okay. Yes. This. Yes. Hmm? Okay. Okay. Change the battery and things. Okay. No problem.
Good afternoon.
Okay.
This is next. This is there. Okay. Good afternoon. And I again thank you, organizer, for inviting me. So, my talk, the perspective and challenges in drug research. Design and synthesis of nitrogen heterocycle as novel therapeutic agents. So, as you know, in drug research, we have three important components. First, lead generation, because if you have no lead, then how you optimize your active molecules. And final component is again very important, that is the process optimization. if you have active molecules and economical viability is not there, then your compound cannot be beat in market and nobody can market your compound. So here the process chemistry start. And here you can see, here the number is greater and here finally number is become very small. But in terms of value, the value is greater. And in morning I already told, drug research involve almost 12 years time. Drug research, this is lab scale, then preclinical, again in lab, clinical trial, phase 1, 2 and 3, evaluation and approval, it took 2 years time, and phase 4 study 2 years. So this is the actual, the picture of drug discovery process. Out of 10,000 compound, 250 compounds, we can say the active molecules. And out of 250 compounds, only 5 compound test compound. Again, out of 5, only 1 compound goes to market and 4 compound due to toxicity or other reason they dropped. This is very interesting slide. Before 1980, we directly inject the natural product in vivo in animals. And after getting activity, we optimize the lead optimization take place. But now the situation is changed. Due to animal ethics problem and other problem, you cannot inject your molecule direct in animal. So now we know several basic biochemical and physiological studies there. We know the genomics and proteomics. And we know the several targets. We synthesize the compound against the specific target. And with the specific target, we synthesize the molecule as active molecules. The first example of this type molecule is captopril. This is the first compound developed by Bristol Myers Squibb, 1981, using the target-based synthesis of the molecules. And this is again the very good information. 74% compounds belong to chemical classes. So you can understand the role of chemist in drug development is greater. Here again the very important slide, as I already told, the natural product, they having the great contribution. Out of 766 compound, the natural product having 249 compounds there from the natural product origin. So this is the overall contribution. I already told around 250,000 or 350,000 plant species identified. Out of only 35,000 compound is, we can say, having the biological activity. So I will not go in detail the morphine and other compound developed earlier stage. So here our institute. This is the Chhatar Manzil Palace. And this Central Drug Research Institute, only organization, those having the capability to develop compound from lab to market stage. So this is the importance of this compound. The 1951, the Jawaharlal Nehru inaugurated this institute, and at that time the skilled manpower is a great problem for pharma industry. This, due to existence of Central Drug Research Institute, our trained manpower in pharma industry, they found there is no shortage, and still in most of pharma industry, the most of president and CEOs, our alumni. This is the contribution, the CDRI. This all compound is marketed. Centroman, this is contraceptive. Keen Mind, this is the memory enhancer. Alpha Beta Archeater, this is the anti-malarial. So all these compound is marketed by the CDRI. This is the Centroman. Recently, Ministry of Health and Welfare introduced Saheli as a contraceptive agent in national family planning. So this is a very important. What the strategy we adopt to find the active molecules? So there are so many strategy, combination therapy, or modification in existing drug. Or you can synthesize the hybrid molecules. In morning, the Diwan Singh discussed about some hybrid molecules. So I will discuss only anti-parasitic, my contribution in anti-parasitic, especially synthesis of anti-malarial compound and anti-leishmanial compound only. This figure is very important to know. 1,300 compound developed for all indication between 1975 and 1999. Only 13 were from tropical disease. So this is very important to know why we have no drug against the tropical disease. The reason behind, because tropical disease of the poor people, poor country, developing country. For multinational, this is not good idea to invest in tropical disease, because patient is poor, they cannot buy the drug, expensive drug. So work in tropical disease is very important to protect the life of the poor people. So I will not go in detail in these. These are known drugs available in market. Still we require new drug because the resistance is great problem. So second line drug we require. For this we need new molecules. This is again very important to know. First time in history of Nobel Prize, no scientist belong from tropical disease area receive any type of award from Nobel Committee. First time they recognize the contribution of the scientists who work on malaria and filaria, and they received the Nobel Prize on their contribution. This is the artemisinin, and other is the development of ivermectin. So you can visualize those scientists work in tropical disease, they have never have any kind of recognition from any committee, any award, because this type of disease is poor people, and they understand that these people, poor people work, they no recognition. If work in cancer and other area, you will find the more possibility to getting the award. This is the fact of the present scenario. So I will not go detail. I will just tell you how we get the develop the synthetic molecules. So there is several approaches. Communal approach, we synthesize the several molecules. As you know, to synthesize the molecules in faster manner, this combinatorial chemistry is very good. In one day, we can synthesize 96 compounds in single day using the organic synthesizer. And those compounds you can test with a high throughput screen, and you can identify the active molecules. So this type of molecules we have developed and published. So this is again the scheme of the solid support. I will not go detail. Lekin the problem in solid support or combinatorial chemistry, you cannot synthesize a compound in kilo scale. This is the limitation, and more of time of duration of reaction is greater. So now we synthesize the molecule hybrid approach. As in morning, Diwan Singh discussed about the hybrid approach. So I will just discuss what the hybrid approach. So this is the classification of hybrid molecules. Conjugate hybrids, here metabolically stable linker, but both having the different target site. And second type of cleavage, conjugate hybrid. So these are 4 types of hybrid molecules you can synthesize. So I will not go in detail. So this is the first example of synthesis of hybrid molecules. Here artemisinin, they combine in quinine and synthesize the artemisinin quinine class of molecules. And what they found, these compound is better than the starting compound. So here the idea of hybrid molecule started, and these trioxane again the hybrid molecules. So we have synthesized the several hybrid molecules, where here we have to intact the 4-amino 7-chloroquinine intact, because this moiety is very important for hematine, to inhibit the hematine polymerization, and the side chain we changed. And here we published the trioxane, the review hybrid molecules for the treatment of malaria, drug news perspective 2010. This review article is highly cited. So this is all hybrid molecules we have synthesized. I will not go detail, but only tell in hybrid molecules you have to keep the 4-amino moiety is intact, and you have to play with side chain only. Here again you can see this is intact, and beta carbonyl class of hybrid molecules we have synthesized. So this is the several publications we have published. I will not discuss. So this is again the hybrid molecules, different type of molecules. Here we side chain introduce here pyrimidine class of molecules, because pyrimidine also having very good anti-malarial activity. So we thought if we hybridize with pyrimidine with this class side chain, so what happens? So this is, although these compounds have shown and not very promising activity. So this is again the trizine class of molecules. This is analog of the cycloguanyl type of molecules. 19 compounds we have synthesized, and this is a 50 most cited article published in VMCL 2005. Similarly, we replace the alkyl side chain with aromatic, and these compounds again have shown very good activity. So just I am giving some idea how we hybridize the molecules, but in all molecules you have to keep intact the 4-amino 7-chloro 4-aminoquinine intact. So these molecules is very active and one of the most active molecules, and they have shown activity against the resistant strain. So these two is all these four molecules shown very good activity against resistant strain. Again, these are different modification is there, and most of compound is more active than the chloroquine. So I will discuss some work our leishmaniasis. Leishmaniasis having the only four compounds available in market. These are pentamidine and sodium stibukogluconate, miltefosine, and amphotericin B. So we hybridize these molecules here. This is the pentamidine moiety and hybridized with the application of sin. This is the natural product. And this compound found very active against the Leishmania parasite. And this paper we have published, Journal of Medicinal Chemistry. So all these type of modification we have done, and most of compounds shown very good activity against the pentamidine. This is again the very good class of compound, quinolidine non-scaffold, and most of compounds shown in vitro more active than the standard drug. So these again molecules, I will not go in detail. So these are most active compounds and have shown in vivo activity 50 milligram per kg into 5 days IP route. So again, this is one of the most. Here we change the side chain amidine and keeping intact the pentamidine skeleton here. So here one of thing is very important, how to develop new prototype in anti-malarial or anti-leishmanial activity. For this we use the multi-component reaction and isocyanide based multi-component, and this is the advantage of the multi-component reaction. We have synthesized the benzoxazepine class of molecules. So this is our protocol, and this paper we have published, Organic and Biomolecular Chemistry. So all these prototypes we have synthesized using the multi-component reaction, and most of paper published in Medicinal Chemistry Communication, Organic Letters, Synlet, RSC Advanced, GOC, Tetraeden Letters, Journal of Organic Chemistry. So here the drug discovery process continuously being transformed by the availability of new tools, new knowledge in biological and chemical computational science, development of molecules with dual mode of action is a milestone in field of drug discovery. So, but rationally designed molecule can help getting the active molecules. You just randomly synthesize the molecules, they will not help getting the active molecules. And finally, this is very important, ask the right questions and nature will open the doors to her secret. So this is our group, and my acknowledgement to my student. So here I will take some time As you know, I am also General Secretary of Indian Society Chemist and Biologist, and we going to organize 28th ISB International Conference, 8 to 10 January, Marwadi University, Rajkot. So we invite you all to participate. This is our glimpse of conference, some old photograph. And here I will just give the contribution of the society. And you can see the most of places we organize our Indian Society Chemist Biologist Conference. If we think in numbers, till date, 26,000 attendees at the ICB conference. Means 27 years of existence of ISCB, 26,000 participants attended our conference. 3,500 life members and 1,500 is the annual member. 5,000 membership we having for Indian Society Chemist and Biologist. And how we help the university to getting the postdoctoral fellow. We also placed almost more than 100 people in industry and placed those our students, very good organization. We also publishing Chemistry and Biology Interface Journal, and 75 issue already published and available on website. 400 paper published in Chemistry and Biology Interface. So this is again the glimpse we invited GIRFS, Center Derivation Institute, and this is Nancy Jackson, President of American Chemical Society, and this is Cynthia Burrows, Associate Editor of Journal of Organic Chemistry. And this is my group with Professor GIRFS. So with this few words, I again thank you to all. Thanks.
So the talk is open for discussion. If you have any query or doubt, you may ask. Professor Chauhan is the right person to deal with drug development.
Actually, I have done it. No question I want to learn actually. Being organic chemist, like the suppression of already existing drugs. So what exactly happens when some either it is a change in the drug molecule will be the same, right? So change in the what you call pathogen or what you call in their morphology or some what kind of mechanism. I mean, what basically it happens like so that one drug already existing drug gets registered.
Actually,
Sir, please
as we also,
come here.
human being also want to survive and parasite also want to survive. So when you inject the drug very frequently, the nature allow the parasite to get resist against that parasite, and they incorporate that drug and use that drug as a feeding material. So for this you require some kind of drug that is new for parasite. So this is very layman answer. Maybe this help you. Okay.
Any other question? If no question, let us thank Professor Chauhan for his very nice presentation. Thank you, sir. Sir, you please collect the memento. You can't go without that. Huh? That is proof.
Thanks.
this is a short break for tea. We will start next session just after 10 minutes because we are already late. So I request you all, please go and take a cup of tea and come with tea here.